My name is Qazi. I am a Mphil scholar, second semester, and this seminar is presented to Dr. Niyaz Muhammad, Associate Professor, Microbiology Department, CUST. And the research or topic that I am going to present today is a novel Acinetobacter baumannii bacteriophage endolysine (LYS AB54) with high antibacterial activity against multiple gram-negative microbes. The outline of the research articles are introduction, materials and methods, results, discussion, and references at the end. I will start my research article presentation from introduction. Acinetobacter baumannii is a opportunistic gram-negative pathogen that can initiate several serious nosocomial infections, which majorly includes wound infection, urinary tract infection, ventilator-related pneumonia, and secondary meningitis. The high resistance of Acinetobacter baumannii towards conventional antibiotics makes their management very cumbersome. Opportunistic pathogens are those which require opportunity to become pathogenic. They are not really pathogenic, but whenever there is an opportunity available, they become pathogenic. And nosocomial infections are hospital-related infections that are acquired from the hospital environment. Among various available alternative agents, bacteriophage encoded bacterioglycan hydrolysis, popularly known as endolysine or lysine, represent a promising alternative for the treatment of drug-resistant bacteria. Endolysine are enzymatic proteins that are produced inside the bacterial host at the later stage of bacteriophage replication and can lyse bacterial host when applied externally. Endolysine renders numerous advantages over the alternative antibacterial agents, including high activity against drug-resistant pathogens, low chances of resistance, and high specificity toward the target bacteria. Treating bacteria with outer membrane permeabilizers such as citric acid, trichloromethane, Triton X100, and EDTA can improve the antibacterial activity of endolysine and conquer with the hindrance presented by the outer membrane of gram-negative bacteria. Material and methods: collection of water sample for bacteriophage isolation. The water sample was collected in the sterile 50 ml screw tubes from the Tongji Hospital, Wuhan, Hubei Province, China. Isolation of bacteriophage P54. The Acinetobacter baumannii strain WHG3083 was cultured in lysogeny broth at 37 degrees centigrade to logarithmic phase and then transferred to the conical flask. Conical flask solution containing water sample, phage buffer, and bacteria were incubated aerobically in shaking incubator on 160 rpm round per minute at 37 degrees centigrade for 2 to 3 days. After incubation, solution was centrifuged 1000 rpm for 10 minutes, 4 degrees centigrade, and filtered through a sterile 0.22 micrometer syringe filter. Bacteria has four growth phases: lag phase, log phase, stationary phase, and death phase. Then 500 ml of the filtrate was mixed with 500 ml of Acinetobacter baumannii in a sterile 1.5 ml microcentrifuge tube and incubated aerobically in a shaking incubator on 160 rpm at 37 degrees centigrade for 15 to 20 minutes. The mixture was mixed with 4 ml of 0.7% soft agar, approximately 50 degrees centigrade, in a sterile 15 ml tube, and poured onto the LB agar plates and incubated overnight at 37 degrees centigrade. Purification of the bacteriophage P54. The microcentrifuge tube containing flake was vortexed for approximately 2 to 3 minutes. The vortex solution was further filtered through a sterile 0.22 micrometer syringe filter. 10 ml of the filter solution, 10 for fold serially diluted was mixed with the 1 ml of Acinetobacter baumannii cell suspension and incubated on 160 rpm at 37 degrees centigrade for 15 to 30 minutes. Afterwards, bacterium phage mixture was mixed with 4 ml of soft agar and poured onto LB agar plates and incubated overnight at 37 degrees centigrade. This cycle was repeated for 5 times to obtain purified bacteriophage P54. Bacteriophage P54 genome extraction and sequencing analysis. The bacteriophage P54 genome was extracted and sequenced following the protocol and procedure. The sequenced genome was compared with available whole genome sequences of Acinetobacter baumannii bacteriophage by the neighbor joining method. Transmission electron microscopy of the bacteriophage P54. The concentrated bacteriophage was stained with freshly prepared phospho-tangestic acid solution 2% for 3 minutes and spot dried on the copper grid for 2 hours. The sample was examined on H7000 FA transmission electron microscope, Hitachi, Japan. Bacterial culture conditions. All bacterial strains were cultured in LB broth aerobically at 37 degrees centigrade. The stocks of bacterial strains containing 20% cholesterol was stored in -80 degrees centigrade refrigerator for long time storage. E. coli, BL21, PE3, Takara cells were used for cloning and protein expression. Cloning of LYS-AB54 in the expression vector. The open reading frame encoding the putative endolysin LYS-AB54 was amplified by polymerase chain reaction PCR with the primers 5 to 3, 54 forward, Ancol, and 54 reverse, Exol. PCR product was digested with precision endonuclease enzymes Ancol and Exol thermoscientific. Purified PCR product was degraded into PET28A positive and transformed to the competent E. coli BL21DE3 cells. The inserted gene was further confirmed by Sanger sequencing. Protein expression and purification. E. coli BL21DE3 containing the PET28A LYS AB54 was grown in 500 ml LB with 100 mg per ml of kanamycin for approximately 2 to 3 hours until the OD optical density 600 reached to 0.5 to 0.6. Protein expression was induced with 1 millimolar isopropyl beta D thiogalactopyronicide at 16 degrees centigrade for 16 hours. The bacterial culture was harvested by centrifugation at 10,000 rpm at 4 degrees centigrade for 10 minutes. Bacterial cells were washed once by PBS pH 7.4 and centrifuged at 10,000 rpm at 4 degrees centigrade for 10 minutes. Cells were disrupted through sonication ice-cold environment. Sonication is a procedure of applying sound energy to excited particles in solution. The insoluble cell fragments were removed through centrifugation at 10,000 rpm at 4 degrees centigrade for 10 minutes, and supernatant was collected by filter through 0.22 micrometer syringe filter. His tag LYS-AB54 was purified using nickel nitriloacetic acid affinity column chromatography with gradient of imidazole solution. After purification, protein were dialyzed overnight at 4 degrees centigrade against trace buffer pH 7.4. The purity of protein was assessed using SDSP, sodium dodecyl sulfate polychromate gel electrophoresis is a discontinuous electrophoretic system which is commonly used as a method to separate proteins with molecular masses between 5 and 2 to 50 kilodalton. Antibacterial activity assay of endolysin LYS-EB54 to determine the antibacterial activity of LYS-EB54. The bacteria were cultured in LB broth at 37 degrees centigrade for both the logarithmic phase OD 600 is equal to 0.6 to 0.8 and stationary phase OD 600 is equal to 1.4 to 1.6. Bacterial culture was centrifuged at 8000 rpm at 4 degrees centigrade for 5 minutes, and bacterial cells were washed and resuspended at risk of pH 7.4. LYS-EB54 was added into bacterial suspension and incubated aerobically at 37 degrees centigrade for 60 minutes. Groups treated with the same amount of tris buffer instead of LYS-EB54 were used as negative controls. The mixture was then serially diluted, then plated on the LB agar petri dishes, incubated overnight aerobically at 37 degrees centigrade. Killing range of LYS-EB54 to evaluate the bactericidal activity of endolysin LYS-EB54 against clinical gram-negative isolate logarithmic phase. Bacteria were treated with 100 mg per ml of LYS-EB54 at 37 degrees centigrade for 1 hour. The reduction in bacterial count in terms of log CFU per ml was defined as the antibacterial activity of LYS-EB54 against tested isolate. The activity of LYS-EB54 in complex media. To test the antibacterial activity of endolysin LYS-EB54 against Acinetobacter baumannii in complex medium logarithmic phase, bacteria were resuspended in LB broth and human serum and treated with 100 mg per ml of LYS-EB54 at 37 degrees centigrade for 1 hour. The mixture was severely diluted and plated on LB agar petri dishes for viable cell count. Statistical analysis. The data were analyzed using GraphPad Prism version 5 software on Microsoft Windows 2019 operating system. The data was expressed in terms of mean positive negative standard deviation (SD) for statistical considerations. Results. Characterization of bacteriophage P54 double-layer agar assay showed clear plaques on Acinetobacter baumannii lawn, postulating the lytic potential of isolated bacteriophage P54. Based on these observations, bacteriophage P54 can be classified to the family of Myoviridae according to the test classification by the International Committee on Taxonomy of Virus (ICTV). Genome sequencing and ORF (Open Reading Frame) annotation of bacteriophage P54. Full genome sequence analysis showed that bacteriophage P54 consists of 1,65,813 base pair with GC content of 36.3%. A BLAST-P analysis further revealed that bacteriophage P54 contained 248 predicted open reading frames with putative functions categorized into multiple function modules, including phage structure, host lysis, phage DNA, metabolism, and modifications. Bioinformatics analysis of endolysin LYS-EB54. A phylogenetic analysis of LYS-EB54 and another gram-negative endolysin showed that LYS-EB54 has a similarity with other reported endolysin-containing lysozyme domains such as LYS-P826 of Acinetobacter baumannii phage. Prior-to server-based structural prediction showed that LYS-EB54 contains multiple alpha helix domains with the absence of predictable beta-plated sheets. LYS-EB54 shows high bactericidal activity against Acinetobacter baumannii. The time-killing assay showed that the LYS-EB54 100 mg per ml would kill logarithmic Acinetobacter baumannii with 0.6 logs of reduction in the first 1 minute of incubation, while up to 4 logs of decrement in bacteria count was achieved after incubation for 10 minutes. Antibacterial activity of LYS-EB54 against logarithmic phase gram-negative bacteria. LYS-EB54 showed high bactericidal activity against all isolates of Acinetobacter baumannii, E. coli, and Klebsiella pneumoniae tested. Moreover, 8 out of 10 Pseudomonas aeruginosa strains were found to be highly susceptible to LYS-EB54. The difference in antibacterial activity of log reduction against these clinical strains may be due to difference in the molecular architecture of the bacterial outer membrane. Notably, reduction of 4 logs from 4.2 to 0, 2.17 logs from 5.77 to 3.60, 2 logs from 4.22 to 2.16, and 2.33 log from 3.86 to 1.53 were observed in Acinetobacter baumannii WHG 40090, Pseudomonas aeruginosa WHG 50023, E. coli WHG 11023, and Klebsiella pneumoniae WHG 11004 respectively. Antibacterial activities of LYS-EB54 against logarithmic phase of gram-negative microbes in tris buffer logarithmic phase strains of Acinetobacter baumannii, graph A, Pseudomonas aeruginosa, graph B, E. coli, graph C, and Klebsiella pneumoniae, graph D, were treated with 100 mg per ml of LYS-EB54 for 1 hour at 37 degrees centigrade. The viable cell number was calculated on the LB aggregates. Data are shown as a mean, positive, negative standard deviation. Antibacterial activity of LYS-EB54 under various conditions. Therefore, we first evaluated the bactericidal activity of LYS-EB54 with the bacteria under different growth phases. Results showed that LYS-EB54 showed similar antibacterial activity against both logarithmic phase as well as stationary phase of Acinetobacter baumannii. These results indicated that bacteria face a minor effect on the antibacterial activity of LYS-EB54. Most of the reported endolysine loses their activity in serum. In this context, we checked out the antibacterial activity of LYS-EB54 against the logarithmic phase of Acinetobacter baumannii in complex media, including LB broth and human serum. As reported previously, insignificant bactericidal activity of LYS-EB54 was observed in complex media. The possible reason for loss of bactericidal activity is the ion exchange in these complex media, which can potentially neutralize the interaction between endolysine and the negatively charged outer membrane. Discussion. Due to its intrinsic resistance, the overuse of antibiotics, some isolates of Acinetobacter baumannii are now resistant to almost all known antibiotics. Indolysine is a class of novel antimicrobial agents to treat and cure antibiotic-resistant bacteria due to its rapid bactericidal activity and low chances of antibiotic resistance. One excellent merit of the LYS-EB54 is its robust bactericidal activity against both logarithmic and stationary phase of gram-negative bacteria in the absence of OMPs, outer membrane permeabilizers. Encouragingly, a reduction over 4 logs in viable bacterial number number was observed after treatment of infected pulmonary cells with 100 mg per ml of LYS AB54 for 10 minutes. Such lysines with broad host range are commonly contain a lysozyme or glycoside hydrolase catalytic tree which cleaves the B1,4-glycosidic linkage between N-acetyl muramic acid (NAM) and N-acetylglucosamine in peptidoglycans that is generally shared by various bacteria. Our results showed that LYS-AB54 completely loses its antibacterial activity in the complex medium like algae broth and serum, which narrow down its application on tropical infections such as bunwound or sort infections. Or possible reason for the loss of LYS-AB54 activity in these media may be due to ion exchanging of endolysine in these environments, which could possibly neutralize the antibacterial activity of endolysines. The OMP-independent activity of LYS-AB54 makes it a potential candidate for the treatment of infection caused by multiple drug-resistant gram-negative pathogens. References. Here are some of the references from the article. Here are some more references from the article. Thank you for your attention.